Hi everyone, welcome to our Biology 110 video presentation on measuring lactase enzymatic activity. In this presentation, I will first introduce some background information and basic knowledge about enzyme and the relevant principles in this lab. Then I will go through the steps of the enzyme experiment, showing how to research the relationship between reaction time and the enzyme efficiency. In this lab, we mainly focus on the hydrolysis of lactose by lactase to produce beta-D galactose and beta-D glucose. At last, I will talk about the real-world connection, including some application of enzyme in daily life. Now I will start talking about background information of enzyme. First, what is enzyme? Enzymes are biological catalysts that help speed up metabolism or the chemical reactions in our bodies. They build some substances and break others down. They are highly specific to certain substrates. In most cases, enzymes are proteins. A large protein enzyme molecule is composed of one or more amino acid chains called polypeptide chains. The amino acid sequence determines the characteristic folding patterns of the protein's structure, which is essential to the enzyme specificity. Few enzymes are nucleic acids called catalytic RNA. And enzymes have remarkable power, which can increase chemical reaction rates by at least 1 million-fold. After the reaction, the enzyme keeps unchanged. Enzymes function best under certain temperature and pH. Enzymes work best when your body temperature is normal at around 37 degrees Celsius. Too high or too low temperature would slow down certain chemical reactions. This is why a high fever would disrupt normal body functions. Each enzyme has an optimal pH. The picture in the slide shows that pepsin functions best in an acid environment, while trypsin tends to function best in a neutral range. Next, we will talk about how enzymes work. The enzyme catalyzed reaction takes place in the inner region of the enzyme called active site. The active sites are very specific, which is only react with certain types of substrates. When certain substrates combine with the enzyme in the active site, enzyme-substrate complex forms. During the reaction, the substrates are either decomposed or synthesized. In this way, new products are formed. Apart from what I mentioned above, enzyme activities can be inhibited in various ways. It can be divided into competitive inhibition and non-competitive inhibition. Competitive inhibition occurs when the molecules very similar to the substrate molecules bind to the active site and prevent binding of the actual substrate. Non-competitive inhibition occurs when an inhibitor binds to the enzyme at a location other than the active site, but it still affects normal function of the enzyme. Next, we will talk about the lab experiment. The enzyme of choice in this lab is glycosidic hydrolase lactase, which can be extracted from dietary supplement tablets. When people drink milk, there is lactase to break down lactose so people can digest it better. Lactase catalyzes the hydrolysis of the beta-glycosidic bond in the disaccharide lactose, releasing lactose and glucose. However, people who are lactose intolerant don't have that enzyme or simply don't have enough of it. In this way, they might have a harder time digesting it. As we want to explore the relationship between reaction time and enzyme efficiency by getting relevant data and plotting a graph, we use spectral photometry to measure lactase enzymatic activity under different conditions. In this lab, the underlying principle of UV spectral photometer is that different substances absorb different wavelengths of light. One substance has its unique absorption spectrum. Therefore, a solution with a certain concentration absorbs some wavelength of light. Correspondingly, some wavelength of light would transmit through the solution with a certain concentration. In the measurement of UV spectral photometer, transmittance is usually converted into absorbance that is proportional to the solute concentration. Based on this principle, either the substrate or the product in this lab should absorb visible light. However, none of the molecules in enzymatic action of lactose can do. We chose OMPG with beta-glycosidic linkage as the substrate in this lab. OMPG mimics the shape of lactose and can be hydrolyzed by lactase as well. The basic chemical reaction is shown in the picture. Additional materials we need in this lab include mortar and pestle, a tube of PBS buffer, a tube of sodium carbonate, and 3 pipettes with a range of P20, P200, and P1000 and pipette tips, 17 microcentrifuge tubes, vortex, a dry bath at 37 degrees Celsius, and 196 well plate. During the enzyme preparation phase, we crush 1 lactase tablet containing 200 milligram of enzyme using a mortar and pestle. Then we put the powder onto a folded paper and transfer it to a 15 ml tube as suspension. After it, we add 10 ml PBS buffer to the tube. Then we put them on the box for 1 minute to make them mix together. The next step is to transfer 1 ml suspension into 2 tubes dividedly and centrifuge them for 1 minute. Finally, we transfer 500 ml supernatant from each tube to a new microcentrifuge tube. In this way, we get lactase extract. Then is the reaction phase. To measure the rate of the reaction, we make measurements of the product concentration over a period of time. We set up 4 reactions and each reaction will repeat 3 times. The 4 reactions were stopped by sodium carbonate at 30 seconds, 60 seconds, 90 seconds, and 120 seconds. We prepare 14 microcentrifuge tubes, labeled as the names in the table. Then we add corresponding PBS buffer, OMPG, and lactase extract to each tube. Once lactase extracted is added, tap the tube to mix it and immediately position it in the dry bath at 37 degrees Celsius for desired time. When the time is up, immediately take out the tube and add 500 ml of sodium carbonate to stop the reaction. Repeat the processes mentioned above for all other tubes in the table. Finally, pipette 200 ml from each tube to the 96 well plate and measure the absorbance at 450 nm. We can analyze the data from microplate reader like the table shown in the slide and correspondingly draw a curve showing the relationship between absorbance and the reaction time. Now we'll talk about the real-world connection. First is the washing powder. Enzymes are universally applied in modern detergent products as they break down soils and stains. The major types of enzymes used are proteases to remove proteinaceous stains, alpha amylases for food stain removal, and lipases for fat stain removal. Second, enzymes are also applied in paper production. It can remove ink, treat starches for paper applications, which enhances bleaching or improve the softness of tissues. In these cases, enzymes bind with the target substance and react. In this way, the target substance can be removed with the production of new substances. Enzymes are widely used in the food industry as well to process raw materials for many products such as dairy, bakery products, meat products, fruit products, beer, and wine. That's all for my presentation. Here is my reference list. Before the end of this video, I appreciate Professor Lee a lot for his patient teaching on biology introduction. Through this course, I gradually have a deeper understanding of cell structure and function, gene expression, evolution, and so on. Additionally, much thanks to Qingling, Ruiqi, and Zhengshan in our TPS group. They actively shared their ideas on TPS problems and explained many points to me when I had some questions. Also, thank you very much for watching my presentation video.